Well, hello, my fellow pre-meds. Welcome back to the second part of our discussion on DNA. In the first slide deck, we talked about structure and organization. In this one, we're going to talk about replication and repair. Be sure to watch that video first because there are concepts that you need to understand from that video that we will build upon in this slide deck. And guys, not only is this topic extremely high yield, but it really spans across multiple topics that we'll see again and again, such as in the lab technique playlist. So definitely try to nail these points down. Did you know that every single video has customized Anki cards associated with it that cover the concepts that we discuss in a video? So if you want a way to be able to actively review the material and retain it for the long run, I've got you covered. If you're interested in accessing the Anki cards or even the slide decks within the videos, then check out our Patreon or reach out to me directly for alternative ways to access the Anki cards and the slide deck. Links in the description down below, as well as the links for everything else. All right, so let's get started right here. We're going to first begin by discussing the origins of replication and the replication fork. So the origin of replication is essentially where you start replicating. So if you look at this, you can imagine that this was once just a linear double-stranded DNA. In reality, it's helical, but just imagine it as a linear structure over here. And then you get these areas where helicase, we'll talk more about helicase in later slides, separates the two strands from one another, and then you get your origin of replication. You got another one over here. Because this is a eukaryotic DNA, it has multiple origin sites, whereas if you look at the prokaryotic DNA, you'll see that there's only one origin of replication site, and that's because DNA in eukaryotic cells is a lot longer, and so you want the process to be more efficient, so you have multiple origins of replication. Now, the replication fork is what we refer to as this Y-shaped structure over here. So regardless of how many origins of replication you have, each origin of replication is going to be bidirectional. So you can see that it's going in this direction and in this direction over here. And then if you look at the prokaryotic cell, same thing, you have it's going in both directions. All right, and then this is just showing you after the DNA double strand has fully been replicated, you'll have two sister chromatids that are joined at centromeres, which we'll talk more about when we talk about mitosis in our biology playlist, but essentially these are going to separate during mitosis. In the case of prokaryotic cell DNA replication, you'll see that the two circular DNAs just completely separate and they don't remain joined at that centromere point. Okay, so now let's move on and talk about the directionality of DNA replication. We talked a bit about the directionality of the DNA backbone itself in the previous slide deck, but I'll quickly review it over here with our analogy, and then we'll talk about how the implications it has for DNA replication. So you can think of this highway as literally the double strand. So I'll show you how it's analogous. So if you're standing on a bridge on top of a highway and you're observing traffic, you'll see that one side of the highway is going to be driving away from you, in which case you'll see red lights because you're going to be seeing the brake lights of those cars. And then the other side is approaching you, right? So what you're going to see is whitish yellow lights or like bluish white lights because those are the headlights, all right? So if you were to think about it logically, where would you be able to see new cars joining? Well, you can see new cars joining the highway on this side from here, but it would be a disaster, of course, if new cars were joining from here. So they're actually going to join from that end. And this is exactly analogous to what's happening in DNA. So notice how DNA, again, has that directionality to it. So it has the 5' to 3' end and the 3' to 5'. So essentially the double helix, the strands are running anti-parallel to one another. And so you can think of this 5' end when you're looking, if you're standing right here and you're looking at the double helix, this 5' end is basically those headlights, and the 3' end is the brake lights. And so again, going back to our analogy, you can only add new base pairs in the case of replication, as we'll see right now, to the 3' end. So you can either add them over here or here, but you cannot add them to the 5' end. That's just the nature of how DNA polymerase works. So let's take a look at at how this applies to replication. So you can see right here that this is our DNA before it's been separated by helicase. Once you separate it, you can imagine helicase, and more on helicase in the next slide. So helicase will separate the strands, and then you'll have, so this is your origin of replication. these are the forks, and then we just slice it in half over here, and we'll take it up there to discuss it further. So this is one of our forks over here. Now notice what I have written down here. The leading strand is read from the 3' to the 5' end, and it's synthesized from the 5' to the 3' end. And then the lagging strand is going to be read from the 3' end to the 5' end, and synthesized from the 5' to the 3'. Now notice that's the same exact statement. This is not a typo, that's intentional. I wanted to really hammer that point down, that both the leading and lagging strand are read from 3' to 5' because the outcome, so essentially when you're reading from 3' to 5', you're reading that parent strand. So these are what we call the parent strands. And then the outcome of that process, which is going to be the daughter strand, is going to be in the 5' to 3' direction. And so what we'll notice here is that the leading strand is called leading because it is a continuous strand. So the outcome of this replication process at this strand is going to be a continuous strand. And that's because if you could imagine that helicase started somewhere around here and then continued to unravel the double strand downstream. And well, the DNA polymerase that's starting at the 3' appropriately, it should be starting at the 3', at this end can just continue following helicase along its path of action, right? So it continues going downstream with it. However, when we're looking at the other strand, which we call the lagging strand up there, you're going to see that because DNA polymerase at this end cannot follow helicase, in other words, it can't go in this direction, it's going to have to continuously, as helicase unravels more DNA, it can jump back and synthesize a little more of this strand. And then once helicase unravels a little more, it can jump back there, and so on and so forth. But the outcome of that is that you get a bunch of fragments, which we call Okazaki fragments. Okay, so essentially, even if you didn't understand the mechanism there, the idea is that the leading strand and the lagging strand are both being synthesized in the 5' to 3' direction. And because the leading strand is following the route of helicase, it will result in a continuous strand, and the lagging strand will result in a discontinuous strand. So more on the different enzymes that are involved in this process over here. So that green enzyme over there is helicase, and helicase is going to separate the two strands from one another. All right, so it's right here, but you can imagine that it started somewhere down here where the strands were previously joined to one another, and then it separated them, and now it's going down the strand until it reaches the very end to replicate the DNA. Now you can notice that topoisomerase is an enzyme right here, and topoisomerase's function is to prevent supercoiling of the DNA. And what does this mean? So if you ever used one of these old phones with these sort of cords over here, if they sort of wrapped around, if the rope wrapped around itself and then you tried to separate it from either end, you'll notice that if you pull hard on it, it'll kind of like start to coil up on itself. That's kind of what happens with DNA, and so topoisomerase helps relieve that supercoiling. Sometimes it's also called gyrase, so just be aware of that. Next, we have another thing that helps stabilize the separate strands of DNA are these single strand binding proteins. So they'll bind to the single strand to stabilize it. And then the last thing we'll talk about before we move on to talk about DNA polymerase is primase. So it turns out that DNA polymerase cannot start the process of replication by itself. So in other words, it can't add that first building block. It can't add that first nucleotide. It needs somebody to come lay down at least a few building blocks, a few nucleotides, and then it can start its action. And essentially, primase is going to do that. It's going to add RNA primers to start the replication process, and then DNA polymerase will carry out its function, again building the new strand, the daughter strand, in a complementary manner. Complementary means that A is binding to adenine is binding to thymine, and guanine is binding to cytosine. So the new strand, the new daughter strand, is complementary to the parent strand. Now, once DNA polymerase has carried out its function, this enzyme called RNase will join in. So what RNase is going to do is essentially, when the primase enzyme added those RNA primers are ribonucleotides. So they're the RNA, they're the nucleotides that are associated with RNA. So recall from our previous discussion on the structure and organization of DNA that the difference between the nucleotides involved in RNA and the ones that are involved in DNA is that the ones involved in DNA will be lacking an OH at the 2' carbon atom, okay? Whereas the ones involved in RNA will contain an OH on their 2' carbon atom. If this is a bit confusing, then you should probably check out our previous video on the structure and organization of DNA. So essentially, RNase is going to come remove those nucleotides that are associated with RNA and add the ones that are associated with DNA, because this is, of course, DNA. And then DNA ligase will come and seal any gaps that are left. And specifically at Okazaki fragments, we haven't talked much about Okazaki fragments, so let's turn our attention to the lagging strand over here. So notice how the reason why polymerase, as you can see, is going in a continuous fashion over here is because, again, it's following the path of action of helicase. So you can see that this strand, the 5' end is over here, we'll follow it along all the way down. You'll see that it's 3' down here. So it's running from this end 3' to 5'. Well, that's appropriate for DNA polymerase because it wants to read the strand, the parent strand, from 3' to 5' and synthesize the new strand from 5' to 3'. Makes sense? However, the lagging strand over here, if you were to follow it along 3' all the way to 5' down here, well, now we run into a problem because the parent strand is running 5' to 3', but DNA polymerase wants to read it in the other direction, in the 3' to 5', and that's why, again, as we mentioned, it'll start jumping around. So it'll maybe build up these nucleotides over here. Then as helicase unraveled a little more, it'll jump back over here, build up a little more. As it unravels more, it'll jump back. And that's why you have Okazaki fragments. And it's essentially the same process. You still need to continuously add those RNA primers every time polymerase is jumping back. And so that results in Okazaki fragments, and you have ligase sealing those gaps after the process has finished. Now, the leading strand and lagging strand, we discussed those, I think, pretty in a detailed manner, I think. So I'll just reiterate that the leading strand is continuous, the lagging strand is discontinuous. All right, now let's talk about telomerase and chromosomal ends. So in the previous slide deck, we talked about what telomeres mean. So telomeres are the ends of chromosomes. Now, why they're relevant in the discussion on replication is because these ends, especially the 3' ends of chromosomes, it's not possible for DNA polymerase to replicate them. So technically, if it wasn't for telomerase, every time you go through a replication cycle, you're going to lose some of that genetic code at the 3' ends. You're going to lose some of those telomeres. However, because telomerase has a built-in RNA template, it can go and mitigate the loss of genetic code. So it'll mitigate the loss of chromosomes by essentially replicating those telomeres, which DNA polymerase was not able to. And then you'll find telomerase abundant in cells that are rapidly dividing, and it makes sense. If they're rapidly dividing, they have the potential to lose more genetic code, so they're gonna have more telomerase working, so in stem cells and many cancer cells. All right, now let's talk about the repair of DNA. So we can discuss the repair process into two broad categories or two different buckets. The first bucket is where you have misplacement or the incorporation of the incorrect nucleotide, or the second bucket where you have damage to DNA itself due to environmental factors. So let's start off with the misplacement or mismatch of nucleotides. So obviously DNA polymerase does its job pretty well, but because it's laying out millions of nucleotides, there's always potential for error. But also, it turns out that when an error does occur, DNA polymerase contains an exonuclease, and the exonuclease is running in the 3' to 5' direction. So the way it's working is that DNA polymerase is going 5' to 3'. This exonuclease works 3' to 5', so that as soon as DNA polymerase accidentally lays the wrong nucleotide, so say it puts an adenine in place of a guanine, it will notice it, remove it, and then DNA polymerase can patch it right away and put the correct nucleotide there. But even if certain mistakes, certain failures within the system escape DNA polymerase's proofreading abilities, there is this other group of enzymes and proteins called mismatch repair enzymes and proteins that can go and correct the mistakes, the incorrectly placed nucleotides after replication has occurred. All right, so, and if you have some defects in those mismatch repair enzymes and proteins, you can increase the rate of mutations that can occur, and that makes sense over there. All right, now let's talk about the second bucket, which is the bucket where there's external damage that occurs to the DNA. So for example, the base pairs themselves, the nitrogenous bases, can be oxidized because of environmental factors. They can be deaminated. So for example, cytosine can be deaminated and it'll convert to uracil. Uracil, recall, is involved in RNA and not DNA, so it shouldn't be there. So that's what we call base excision repair, where exonucleases will go take out the base pair that's defected, or the specific base that was defected. Based on the complementary strand, DNA polymerase can read that complementary strand and place a functional base over there. So next I wanted to talk about nucleotide excision repair. So this is more bulky damage that occurs to the structure of DNA that actually distorts the double helix. So for example, you'll see it on the MCAT. I've seen it all the time where they talk about thiamine dimers. So essentially UV radiation damages the DNA in a manner where two thiamine bases next to each other will form this dimer, and that has to be repaired through nucleotide excision repair. And then again, DNA polymerase, based on the other strand, can lay out to the correct and functional nucleotides, and ligase can come and fill in or seal the gap over there. All right, so now let's talk about oncogenes and tumor suppressor genes. So these are pretty straightforward, but they show up on the MCAT. So proto-oncogenes are genes that are involved in the regulation of cellular differentiation or cellular replication. So in a way, they control cell division. Well, when they get mutated, they convert into what we call oncogenes. And oncogenes are going to allow for uncontrolled cellular proliferation and can promote the formation of tumors and cancers. And all you need is a single mutation in one of the alleles, so you have 2 copies of each. If you have just one mutation, it's going to result in the formation of the increased cellular proliferation and potentially cancer, because it's acting in a way where it's going to stimulate the overproduction of cells, or the overproliferation of cells. Contrast this with tumor suppressor genes. Tumor suppressor genes, you can think of them as brakes, like brakes on cellular proliferation, preventing tumor development and cancer development. So if one were to be mutated, you still have the other brake. So in a way, they're recessive, so that you have to have both of them mutated before cellular proliferation occurs and cancer development can potentially occur. So you can think of the proto-oncogenes as the gas pedal. So as soon as you get one mutation over there, it's dominant, so it'll cause the cellular proliferation right away, whereas tumor suppressor genes are like brakes. Okay, and examples of tumor suppressor genes are P53 and RB genes. These are the ones that show up quite frequently on the MCAT, so just be familiar with them. And I think, yeah, that wraps it up. So these are just some knowledge check questions. You can pause the video and try to answer them. Otherwise, I will skip through. And if you have any questions or clarifications about these questions, be sure to hit me up, and I'll be glad to help out. So there's our other question. All right, so that was a very dense slide deck. Very difficult topic to learn, especially the first time around. So if you feel like you need to watch it again, be sure to watch the video again. Also use our Anki cards. They'll really help with memorization. And one note that I didn't mention when we were talking about DNA polymerase is that there is different DNA polymerases between prokaryotic and eukaryotic cells. So for example, in prokaryotic cells, one DNA polymerase that shows up on the MCAT is DNA polymerase 3 in particular. Whereas the ones associated with eukaryotic cells include alpha, delta, gamma. To be honest, I haven't seen them show up on the MCAT, but if you really want to know them, I'll include them in our Anki card slide deck. But again, I think they're very low yield. But yeah, with that being said, if you have any questions, reach out to me. Otherwise, go ahead and crush that MCAT. You got this.